an introduction to Professor Cross for those who are not familiar. So Nick Cross is a professor of human genetics at the University of Southampton, and he also serves as the director of the Wessex Genomics Laboratory Service in Salisbury in the UK. He started his postdoctoral career at the Hammersmith Hospital in London, where he developed an interest in CML under the mentorship of Professor John Goldman. Thus far, his work has resulted in more than 400 peer-reviewed publications. So we're delighted to have Professor Cross with us today to give us the biological overview of CML Highlights of EHA.
Thank you, Nicola. So I've taken a slightly different approach to Andreas, and I've done a lot of abstracts, but with much less detail. So there was some, the scientific presentations were spread across. There was an oral session on Saturday afternoon. There was the ELN EHA scientific working group, and also a small selection of posters as well. And so I've selected some of these and grouped them into 3 themes. So first, prediction of deep molecular response or treatment-free remission. Second, outcome and treatment of blast phase. And third, new therapeutic approaches. So the first I'd like to highlight was an oral presentation from Janine Steichen and Dominic Wolf's group. The novel immune-related score predicts DMR in CML patients. The study was based around a subset of the DASA HIT study that Andreas went over. Blood was drawn at baseline, and there were measurements of immune markers from multi-parameters by using multi-parameter flow cytometry and a multiplex immunoassay. This data was integrated and a predictive model created and tested, and then validated in the NEST first cohort. These are, of course, patients treated with nilotinib. And this is really just a summary slide of the data. So these are the factors that went into the model, a variety of factors with different transform coefficients. Very interestingly, the highest being CD62L, so this is L-selectin, which this group had previously associated with TFR, specifically expression on T cells. Other groups, Agnes Young in Adelaide, has associated expression of this on natural killer cells with outcoming CML. This model predicted MR4 at 24 months. Looking at the cumulative incidence of MR4 and MR5, you can see a clear separation on patients who had a high or low score. And then this is associated also with molecular response in nilotinib-treated patients. So here you see molecular response, MMR, MR4, MR4.5. At different time points, the level of significance the score is predicting this. So further evidence that immunology is important in response to CML. And, you know, we had a lot of studies around immunology that somehow need to be integrated into something that's more specific, but this is potentially a useful and cheap predictor. Second was a machine learning approach to identify a 20-gene transcriptome signature predicting TFR in CML. And this was presented in an oral presentation by Franco Nicolini. Variety of patients included. There were 83 that were basically included in the final study cohort. Blood samples were taken at TKI cessation, and RNA sequencing was performed. And then this cohort was split into a training and testing cohorts. And you can see here that some patients had imatinib, some had nilotinib, and you get roughly the expected frequencies of those who subsequently relapsed or remained in TFR. So this group took a machine learning approach with a custom immune cell deconvolution method, which I don't have time to go into, to identify 27 genes that were differentially expressed between those patients who remained in TFR and those who relapsed. And this then was used to build a predictive signature. So that signature performed well in the training set, obviously, but it was also performed well in the replication cohorts, testing 1 and testing 2. And on multivariate analysis, the score signature here performed well, significant hazard ratios for intermediate and high compared to the low group as a reference. And in this relatively small cohort, there was no influence of MR4 duration or transcript type, factors that are accepted in larger studies as being significantly influencing TFR. So, it looks like this could be a strong signature of TFR. The successful TFR group of enrichments in genes related to glycolysis, reactive oxygen species, the immune cells, immunoproteasome, interferon gamma pathway, whereas the relapse group had an enrichment in genomic instability, senescence, exhausted progenitors, and stemness. So a variety of biological features, but again, with a prominent immune component. There was internal statistical validation, but of course, these results remain to be validated in a prospective setting. And finally, in this section was a presentation by Claire Lucas, again, an oral presentation on Saturday afternoon. So Claire has previously published data showing that the expression of CIP2A as measured by qPCR is predictive of disease progression in chronic phase patients. The new data here was a focus on BCL-XL and TFR. This is just a snapshot of the data. So the study cohort here was a subset of patients from the UK Destiny study. So this is where there was a de-escalation phase prior to stopping. And this referred to 3 samples, one that was taken at the time of initial de-escalation and 2 time points just shortly after that, 3 and 6 months. And this is data in patients who relapsed. And basically you can see these increasing levels of BCL-XL expression, which I haven't got the panel to show here, which you don't see in patients who remain in remission. And this occurs 6 to 8 months before molecular absence detected in the lab, which is quite remarkable. Again, we need more replication data to substantiate these findings, but potentially very interesting. And Claire showed a nice treatment algorithm involving expression analysis of CIP2A and BCL-XL and how that might be integrated into patient management. Okay, the second group of presentations around outcome and treatment of blast phase. And the first, this is in the scientific session, it's arguably a bit more clinical, but it was from Michael Dausiker as part of the ELN and UTOS group, development of a prognostic scoring system for blast crisis, insights from an ELN blast phase registry. So this was based on data from 305 patients, data collected from 12 European countries over quite a long period of time. And these were the factors that came out in the model. Platelets, the phase of disease, this is whether patients presented in blast phase, or whether they presented in chronic phase, then developed blast crisis. The phenotype of the disease, so lymphoid versus other, and of course it's been well known that these are really quite different, with lymphoid having much better prognosis. Presence of extramedullary disease, blasts, and age squared. And when you apply this and to produce a high risk, intermediate risk, and low risk group, the results are really very striking in terms of huge differences in survival between those with a high risk, median of well under a year, and those with a low risk where the median is more than 96 months. So it'll be a while, I think, before this is replicated in the independent cohort. Again, there was a lot of internal statistical replication to ensure the model was robust. And this might provide some hope where you might imagine risk profiling of the blast crisis for treatment in the future. And of course, treatment of blast crisis is extremely challenging. One of many new drugs that is of interest to some people are BH3 mimetics, such as venetoclax. And Mari Copeland gave an excellent presentation in the educational session, which I'd encourage everyone to watch, that mentioned venetoclax. But on the scientific side, I've picked up on 2 abstracts from Katarina Lahova-Polakova's group in Prague. One was a poster which was looking using mass cytometry to predict sensitivity to venetoclax, and a related study that was looking at possible options for asciminib-resistance. And these studies used a common initial strategy to develop cell lines based on KCL22, which is a CML cell line, that were either imatinib-resistant, ponatinib-resistant, or asciminib-resistant. And the schema is showing here. Don't have time to go through it in detail, but basically for asciminib-resistant clones that were selected, there are a variety of BCR-able secondary mutations, plus some other changes which may or may not be relevant. For imatinib-ponatinib-resistant clones, this will start off with T315I, and then a variety of other additional mutations were required. So the poster on venetoclax started with these different clones that were resistant to imatinib and ponatinib, cultured or not with venetoclax, and they developed what they call apoptotic mass cytometry panel to look at 17 different apoptosis-related proteins that are listed here, and where applicable, their phosphorylation states. And there's very interesting correlations. So those patients who had a higher IC50, so they were more resistant to venetoclax compared to those with a lower IC50, you could see clear differences in the amount or the ratio of these different apoptotic proteins. Focusing on individual proteins, marked differences for several of them. Just picking out this caspase, for example, this indicates these green ones are the responders, the sensitive clones. These clones are undergoing apoptosis, and not other differential signals for MYC, AKT, STAT5, and MCL1. And looking at the BCR-ABL activity, and all this is done by the mass cytometry approach, you can see reduction in B-cell activity in those who responded, but not in those cell lines that did not respond. So potentially a predictor of blast crisis that might be responsive to venetoclax, and clearly this will need to be taken into primary patient samples. The other presentation from that group, which was an oral presentation from Nicola Courik, focused on asciminib resistance. And just to go to the sort of bottom line here, this experiment's done in mice. So asciminib resistance is here in this sort of darker red. So the survival median survival is between 10 and 15 days. This is greatly extended if you have ponatinib, but the combination of ponatinib and venetoclax was the most efficacious in this model. And finally, new therapeutic approaches. So I've picked out two here, and these are both from the scientific working group session. The first from Michael Deininger on Protac inhibition of BCR-ABL, and the second from Dave Vitry on the potential for stem cell inhibition. So first of all then, BCR-ABL, we know that the BCR-ABL kinase activity is fundamental to its transforming properties and is essentially what drives CML. It's that activity that's targeted with all the various TKI inhibitors. But the BCR-ABL is a large protein, a complex protein that's known to have other functions, including as a scaffold where it acts as a physical attachment site for other signaling proteins. And there's a variety of data that's reviewed in this paper that's I think now online in Leukemia, will be shortly, indicating the various activities influencing apoptosis, adhesion, migration, repair, and self-renewal, all of which are not dependent on the BCR-ABL kinase element. So how might these be targeted? And related questions, how you effectively target multiply mutated BCR-ABL, the so-called compound mutations. And one possibility is to use these so-called Protacs. These are illustrated here. So a Protac is a tripartite small-ish molecule. So it has a ligand for the protein of interest at one end. So this protein of interest is BCR-ABL, a linker of variable length, and then a ligand for an E3 ligase. So basically, this will then bring together a complex of, in this case, BCR-ABL linked to the E3 ligase. The E3 ligase ubiquitinates BCR-ABL, and BCR-ABL is then degraded by the proteasome. And there are sort of guides for building these things, but a lot of it is empirical as to what E3 is going to work best, and this sort of structure and size of the linker. As for the actual ligand, it's going to be anything that binds ABL BCR. It's going to be standard TKI inhibitors. So my group built a series of these, and just to show a couple of examples here. So just looking at this one, LPA81, you see very efficient degradation of BCR-ABL. These compounds inhibit the growth of BAF3 cells transformed with BCR-ABL. and this LPA81 has the lowest IC50. There are a whole series of controls you need to do to show that these things are working in the way that you expect. So this is a NECAR derivative that won't bind the E3 ligase, and indeed it doesn't degrade BCR-ABL, so that's information that is required. And you put it against a variety of cell lines, including the CML cell lines inhibitor. And just to leap straight to the summary of his talk, so there were two LPA81, 81S derivatives of that capable of selectively degrading both native and mutant BCR-ABL. 81S is more potent than currently available TKIs against BCR-ABL compound mutants. The combination of these Protac with ponatinib stables the inactive conformation, the so-called DFG palette conformation of BCR-ABL compound mutants, and allows for further synergistic activity. So I haven't got the data slide, but the combination of this Protac and ponatinib is particularly active. A lot of work was presented. The inactive conformation of BCR is required for optimal degradation via Protac, and they've now done a particular lysine residue in the ABL-SH2 domain that's required for optimal degradation. So a lot of interesting information. It'll be great to see the study when it's published. And finally, the potential for stem cell inhibition to CML is stem cell reprogramming a more realistic option. So we've seen large numbers of presentations over the years about defining quiescent leukemic stem cells and thinking about how they could be targeted. But it's extremely difficult because these are quiescent cells and they don't express high levels of BCR-ABL. And being quiescent, they don't depend on BCR-ABL. If you can eliminate stem cells, in theory, you can eliminate the disease, but it's an intrinsically difficult problem. So a slightly different angle here. So what Dave's group has done is taken peripheral blood cells from small cohorts of patients who are either good or poor responders to TKI, and done an extensive analysis using single-cell RNA-seq of CD34 plus CD38 negative CML cells. So this is a highly enriched quiescent leukemic stem cell fraction. A lot of data, with the sort of punchlines being that there was a high diversity of molecular subtypes within this fraction, but there were clear differences in these quiescent leukemic stem cells between those patients who responded and those who didn't. So responders are high in TNF, NF-kappa B, and cytokine signaling, while those that failed are high in pathways related to MYC. But if you home in on particular programs involved in lineage commitment, these appear to be different between quiescent leukemic stem cells, between responders and failures. There was particular focus on the MEP lineage. So if you look at the trajectory maps here, this is the kind of trajectory to MEPs in those patients who responded optimally, and a different trajectory in those that didn't. So the question then is, can we use cytokines to reprogram LSCs from TKI failure to become more like LSCs from TKI optimal responders? And can exposure to cytokines improve the sensitivity to TKI? It's a lot of work in both mice and in vitro models. Yeah, I've confused myself with different slides here. Basically, you're looking to do this. Take those patients who are TKI failures, treat with cytokines, reprogram them to become responders. A lot of data, but just to focus on the findings. So yes, exposure of leukemic stem cells from CML patients to cytokines disfavors the failure trajectory and favors this optimal trajectory. Exposure of these cells in PDX mice reprograms them from high MYC, that's failure, to high TNF and NF-kappa B signatures, so that's optimal. And then actual data on bulk cells and exposure of CML stem cells in the PDX model of cytokines improves sensitivity to TKI. So sorry, no time sort of grind through this data in detail, but the conclusion here is that this clearly warrants further research to cytokine-mediated reprogramming that could lead to innovative treatment strategies to target leukemic stem cells and thereby hopefully enhancing patient outcomes.
Thank you, Nick. Thanks. There's obviously so much data and information to try and condense into a 15-minute presentation here. I just apologize to our audience. We've obviously had technical issues with people joining the meeting, so I really appreciate those of you who persisted and were able to join. Obviously, so it's a now small and intimate group, and so it's your opportunity to ask questions of the experts, either of what data that's been shown about EHA highlights or any other questions you have. So I will hand over to the floor to the audience. Tim, did you want to start?
Oh, hi. Yeah, sorry, I came in late because I couldn't get in, so I missed the first half of your talk, Andreas. But I thought the data. Can you hear me?
Yeah,
Yes.
yeah.
Yeah, I thought the data with the Philippe Rousselot study was really interesting. Would you think that's maybe the way to go for your patients who you're not confident?
Yeah, I think it's
given full therapy.
It's one way to go. Yeah, of course, the colleagues in the UK propose high-dose chemotherapy in addition to a tyrosine kinase inhibitor, and I discussed this with Jane already, and she is very strict on that, insisting that all patients who should go to allotransplant should be treated previously with a chemotherapy. We don't do that. We do it very individually. And a patient who is responding to ponatinib alone with blast reduction or even blast elimination, I think it's okay, and we don't need to give him a chemotherapy with morbidity and cytopenia and risk of infection and all this stuff. That means what Rousselot presented here was an in-between. That means epigenetic treatment with TKI. I think it's a good suggestion, but we need, of course, more systematic analysis of that. He had a mix of chronic phase, accelerated phase, and blast crisis patients here. I think when we want to focus on blast crisis, we need more data on this approach.
I think the patients in the chronic phase had also transformed to blast crisis. They
had previous diagnosis. Yeah, that's right. That's right.
Yeah.
They had all. They're
all blast crisis.
Additional chromosomal aberrations. That's right. That's right. That means it was the initial situation. Yeah. Yeah.
Can I just follow that up?
In any case, we should have a subgroup analysis, which patients are eligible for such an approach, which patients should get venetoclax, for instance, which is another approach, and which patients should really get a typical induction chemotherapy.
Can I just follow up? If you just give them ponatinib alone, are you not worried about CNS relapse?
In lymphoid disease, yes.
No, in myeloid, even in myeloid.
Can we avoid this? The question is, can we avoid this with the chemotherapy alone? Yeah,
well,
that's an alternative question. Yeah, it's no, I cannot answer this because
too difficult.
It's limited. Okay. Yeah.
Fair enough.
I've got a question for Nick, but I'll leave it open for other people to ask questions first.
There's nobody with hands raised. I've got one question in the chat, but Tim, you want to ask your question?
Nick, the Protac approach looks really exciting. I probably heard it in your talk and in Mike's, but the specificity, how good is it? Do you also destroy ABL and ABL2, or do you literally only destroy BCR-ABL?
So, I mean, the primary driver of the specificity is that ligand that you put on one end of the Protac. So it's the same question as how specific is imatinib, how specific is lostinib, how specific is ponatinib, whatever you attach.
So, okay, so you're still going to be destroying ABL. You're going to be destroying, depending on which TKI, I can't remember what he actually used. Was it ponatinib in his model?
I can't remember what the final line was. It might have been. But
you'll be hitting other targets then and destroying those proteins as well. I guess so. Toxicity might be a big issue.
Unless you had something that very specifically bound BCR-ABL.
Asciminib, I guess.
Well, yes, and I'm struggling to remember now whether Mike mentioned asciminib and data around that, but that would definitely be one possibility. obviously you've got normal label, is one isoform normal label as well.
Yeah, yeah.
But that in theory would be more specific.
Okay. So one other question.
Okay, I've got one more. Sorry, I've got one more question for Nick. I think the approach that David Vetri was looking at was really elegant and exciting, but gee, it'd be hard to know exactly. I'm not sure he actually mentioned the combination of cytokines that he was using to drive them. Or is that some sort of under IP coverage? So he's not revealing that because
No, it was on,
I imagine.
One of the slides, it was I think it was just IL-3 GM-CSF stem cell factor.
So a standard sort of three factor combination.
Yeah,
Okay.
it was on the slide.
Do you think that's a realistic approach in the future? That, I mean, it might be every patient would have a different cytokine requirement to get them into that sort of, you know, low risk sort of pattern of
I'm
response.
sure it's going to be. I mean, I think the number of patients studied was relatively small. Obviously, this is extremely expensive analysis on a large number of cells, which makes it to expand to a large number of patients. But I think the important thing here is the principle. You know, if the principle is that it could be done,
yeah,
it gives you a route to improving outcomes. Then the challenge is how can that possibly be implemented? I don't think anyone's saying this is going to be something simple.
Not tomorrow's therapy, yeah.
No, but it's at least you feel you're on a path where it's in principle achievable.
Yep.
Okay. So, Andreas, I guess sort of coming back to the clinical part, I mean, were there any questions that came up during the meeting that you think were particularly relevant that could be expanded on here?
No, I think, and Tim is very aware of this question, which is now our major task for the next months and years, is to develop strategies for patients who do not respond optimally to asciminib as a BCR-ABL specific inhibitor. When we start with asciminib and we see a deep molecular response, of course, we want to discontinue and we will test this. But what about these patients who do not respond that fast or complete or whatever? That means which combinations should be tested. I think that's our next approach, and which combination should be tested in which patients at which time point. I think these are the questions which came up. Of course, a lot of people, a lot of colleagues talk about the costs of treatment, but I think, and when we look at oncology in general, any new treatment, any new innovation is expensive, but this is for a certain time only until other treatments are available or the patent is off. That means we live with that in general, and each country should decide what should be available for their patients in the country, and there are established methodologies around. It means I don't see a major point here. And I would try to treat my patients in the most optimal way. That means mainly to achieve the best quality of life. I think that's the major point now. And this was around any discussion around CML treatment, also with the dose optimization studies. It's all about improving quality of life.
Yes, thank you. Well, I think that's an ideal place to end our discussion today. Again, thank you for all of those who were able to join us. We really appreciate the time, Professor Andreas Hockhaus and Professor Nick Cross. Thank you for sharing your expertise, your highlights,